He's currently Vice President of the Oncology Center of Excellence in Drug Discovery at GlaxoSmithKline. He's also an adjunct professor of biochemistry and biophysics at the University of Pennsylvania School of Medicine. Dr. Copeland received his Ph.D. in chemistry from Princeton University, doing pioneering work on Raman spectroscopy on cytochrome C. He continued that work as a postdoc at California Institute of Technology and also in his initial faculty position at University of Chicago School of Medicine in the Department of Biochemistry and Molecular Biology. In the late 1990s, he moved to the pharmaceutical industry with positions at DuPont Merck and at Bristol Myers Squibb, and then assumed his current position at GlaxoSmithKline. He's very involved in drug discovery research, particularly focusing on the cardiovascular system and on cancer. He has over 130 publications and books. And today, the title of his talk is Biochemical Characterization of Kinesin Motor Protein Inhibitors, a Potential Mechanism for Antimitotic Chemotherapy in Cancer. Dr. Copeland.
Thank you very much. I'm very excited to be here, and I thank the organizers for the generous opportunity to speak in such an august company. So I'm going to tell you about a project that we are doing at GSK, which is actually a joint research project with a small biotech company, Cytokinetics, in the San Francisco Bay Area. And this is a project that is a rarity in the pharmaceutical industry because it's actually progressed to the clinic. And the probability of that happening is fairly low. But rather than telling you about the clinical data or a lot of the data that has driven the clinical models, I'm going to focus my attention on the early biochemical characterization of the target and of the interactions of the target with the inhibitor. And I hope what I convince you of today is that that type of detailed mechanistic understanding is a great way to maximize the effectiveness of your drug discovery efforts, because we in the pharmaceutical industry, as you know, are not in the business of doing science. We're in the business of developing medicines. It just so happens that the most effective way of making great medicines is by doing great science. And so that's why we support scientific research. Let me start right up front by acknowledging the folks that played a significant part of this project. This is our project team, and I'd like to, in particular, point out Roman Sakowitz at Cytokinetics, formerly at Cytokinetics, Jeff Carson, and especially very talented scientist Lusong Lo, who directed a lot of the biochemistry in this program. So you may notice that that's a fairly large group of people. And the reason for that is that drug discovery, even the preclinical aspects of drug discovery, is a very complicated matter and requires the combined expertise of a very diverse group of scientific expertise. So I've referred to this as the drug discovery roadmap or the drug discovery project matrix team map. And basically what it does is it gives you sort of a sense of how we go about doing drug discovery in the industry. We start with an unmet medical need. From there, we try to go to some pathological understanding, and from there to a molecular target. And this is really where the rubber meets the road. Once we understand that we have a molecular target that we can go after, that's when we really start to put significant resources on a program. Once we've identified a molecular target that looks interesting, we go through a very detailed stage of target validation basically to ask the question, will pharmacologic modulation of the activity of this target have the phenotypic effects that we expect? Will it be disease modulating? From there, we engage our medicinal chemists and we start to look for lead compounds. And once we have lead compounds, we go through a series of optimization steps onto animal models, safety assessment, and eventually, hopefully, clinical trials. And I've drawn this out as a very linear process, but in fact, it's quite iterative and there's quite a bit of interaction among these different parts of the process. And the main point of this slide is to point out, again, the great diversity of scientific disciplines that have to come together as a team in order to make this work. So the analogy I always like to make is, it doesn't do you any good to be the best shortstop in the American League if you're not playing for the New York Yankees. But no controversy, okay. Okay, this is a snapshot of what we're doing. If I say that in Boston, I get a very different reaction. This is our oncology portfolio. These are some of our approved drugs. These are things that are very close to being approved, and these are some of our earlier stage assets. You can see that it's very rich in things that are inhibiting angiogenesis or inhibiting growth factor signaling, but also we are looking at a number of antimitotics, and the one I'm going to tell you about today is KSP, kinesin spindle protein. And let me start by introducing what KSP does. It's part of a mitotic spindle mechanism for separation of chromosomes during cell division. And so in green you see the microtubules, and the microtubules are involved in cellular transport networks. In the case of mitosis, transport of the chromosomes. But the analogy that Lusong has made, and I will borrow, is that the microtubules are the cellular equivalent of train tracks, and the mitotic kinesins are a family of enzymes which act as cellular trains that transport cargoes along those tracks. And those trains have to be fueled, and they're fueled by ATP hydrolysis. Now, there are a number of drugs that are approved for oncology that act by disrupting microtubule polymerization or depolymerization, the Taxols and the Vincas, and all of these compounds directly bind to and inhibit the microtubules, and as you might expect, that has some unintended consequences. And one of the most severe of the adverse effects that is seen with these type of drugs is peripheral neuropathy. So we thought that by targeting the kinesins in a more specific way, we might be able to avoid some of those toxicities. So there are a number of kinesins that are involved at different stages in mitosis, and KSP, the target that I'll be focusing on right now, is involved very early in spindle pole separation. It's essential for spindle pole function. Its expression profile is consistent with an exclusive role in mitosis. It's overexpressed in a majority of tumor biopsies, and importantly, it's absent from terminally differentiated post-mitotic neurons, so that we might have a mechanism for avoiding some of the adverse effects of the other drugs that are on the market today. So, what do kinesin motor proteins do? They are functional dimers or greater, which consists of two head groups that associate with the microtubules, and there's a stalk and then another set of head groups that binds the cargo. Those cargoes can be a number of different macromolecules. In the case of KSP, the cargo is another microtubule strand. And these head groups are the enzymatic sites of ATP hydrolysis. And if this movie works, what you'll see is that we have ATP binding and hydrolysis, which leads to a hand-over-hand type of epitaxial motion of the microtubules along, of the KSP along the microtubule surface. So we have 2 KSP inhibitors in clinical trials today. The most advanced is this compound, SB715992, which is currently in phase 2 clinical trials. It's a quite potent inhibitor of KSP, sub-nanomolar, about a nanomolar IC50 for inhibiting proliferation in an ovarian tumor cell line, SCOV3s, and a variety of other cell lines, as well as xenograph activity in preclinical models in colon, lung, pancreatic, and a number of other tumor types. I'll mention in passing that not only is this a very potent inhibitor, but it's a very slowly dissociating inhibitor. And that's a topic that's very near and dear to my heart lately, and the topic for an entire other seminar on the importance of drug residence time on targets as a measure of pharmacodynamic potential. So, as I said, for KSP, the protein attaches to 2 strands of microtubules, and under the influence of ATP hydrolysis, moves along the tubules in opposing directions, so that leads to separation of these spindle poles, which is characteristic of mitosis. And so what one would expect is that an inhibitor of KSP would block the separation of these centrosomes and block the separation of the chromosomes. And indeed, that's what one sees. Here, in particular, are the centrosomes, nicely separated in the control cells. They do not separate in the KSP inhibitor-treated cells. And we have some nice movies that take too much time to set up here, where we can show you individual cells where the centrosomes, in the presence of a KSP inhibitor, try to separate and fail, do that a few times, and then the cell says, I can't take this anymore, and apoptosis. So if we think about the chemical pathway of KSP and other mitotic kinesins, we're offered a number of opportunities to engage these enzymes and to inhibit them. So there is a basal ATP hydrolysis activity of the enzyme. The free enzyme binds ATP, forms a binary complex. You get hydrolysis to form ADP. There is a very slow release of ADP to reform the enzyme. That is a very catalytically inefficient process, but in the presence of microtubules, the ATP hydrolysis and especially ADP release, which turns out to be rate limiting, is greatly accelerated. So Kcat over Km is increased about over 1,000-fold in the presence of microtubules. So another common theme to my scientific work in the pharmaceutical industry has been the importance of protein dynamics in enzyme function and enzyme inhibition. And this system is a great example of multiple conformational states along the pathway of reaction that individually could be intervened at with small molecule drugs. The first KSP inhibitor to be reported on was this compound, monastrol, first reported by Mayer et al. In Science. And this compound And the KSP inhibitors that we have in the clinic and all of the KSP inhibitors that have been reported to date, until today, all act by a common mechanism and share a common binding site on the enzyme. And the crystal structure for KSP with monasterol bound has been solved by Yan et al. And monasterol binds to an allosteric site on the enzyme and binds in the presence of ADP. So this is the nucleotide binding site, the center for hydrolysis of ATP, and there's quite a distance between that and the monasterol binding site. And as I said, all of the KSP inhibitors, including our own, bind to this site and behave very similar to monasterol. Now, we know the binding site of monasterol, we know the IC50 for monasterol, and that's sufficient for many people to go into the clinic. And when we looked at the system for monasterol and for all the other KSP inhibitors that were known at the time, there was a conundrum. Monasterol and all of these inhibitors share in common the fact that their cellular potency cannot be accounted for fully by their in vitro potency as KSP inhibitors. And just to exemplify that, for monasterol, the IC50 for inhibiting KSP is reported to be between 50 and 150 micromolar. The cellular EC50 is lower than that, 22 micromolar. And that differential is even greater for some of the compounds that are going into the clinic. So we have more potency in cells than we do against the free enzyme. What's going on here? So the question we ask is why? Why do we ask the question why? Because again, a detailed mechanistic understanding of how our drugs work is the most effective way for us to get to the clinic and to get to the clinic in a way that avoids untoward surprises. And so I've come to tell people in my group that Pasteur once said that chance favors the prepared mind. He got that almost right. That's part of it, but it's really the chance favors the perplexed mind. And so I love it when we're perplexed. I love when we have data that we don't understand like this, when a couple of data points don't quite fit on the line, because that's where the real discoveries come from, is understanding those peculiarities of the data. So we asked the question, why might this be? And the first thing we did was we looked at how we were screening for KSP inhibitors, how the community, GSK as well as others, were doing these enzymatic assays. And we asked the question, what might be different about that from what the enzyme experiences and the inhibitor experiences under physiological conditions? And looking at a variety of different solution conditions, one thing that stood out is that physiological ionic strength is about equivalent to 150 millimolar salt, whereas the KSP assays are all run at 0 salt, essentially no ionic strength. And I asked why that was, and the answer was that, well, it worked. So we screened that way. I just love answers like that. So we said, okay, it's unlikely, but let's take a look at the effect of salt on monasterol interactions with KSP. And here are typical concentration response plots. And indeed, what you see is that as we increase the ionic strength, either with sodium chloride or potassium chloride, we see a salt-dependent diminution of the IC50, an increase in the relative potency of the compounds. Very interesting result, and so we wanted to pursue this further and ask why might salt influence the interactions of monasterol with KSP, because there's nothing about the structure of monasterol and certainly nothing about the structure of the other KSP inhibitors that also show this effect, that would lead one to think that there was some type of electrostatic interaction or other aspect that would be influenced by salt. Now, this is to remind you of what you already know as all good pharmacology students, that in terms of steady state mechanisms, there are basically three types of modalities of enzyme inhibition: competitive, non-competitive, and uncompetitive. And I'll just quickly remind you that by competitive, what we mean are compounds that bind to the same enzyme form as does the substrate, so that substrate and inhibitor binding are mutually exclusive. Non-competitive inhibitors can bind either in the presence or absence of substrate, and uncompetitive inhibitors bind exclusively or almost exclusively to the enzyme substrate complex, or a form that occurs later than the enzyme substrate complex in the catalytic mechanism, but they don't have affinity for the free enzyme. So what about monastrol and these other KSP inhibitors? Well, we looked at the effects of salt on different kinetic parameters of the KSP reaction. And what we found is that over 0 to 150 millimolar KCl, we had essentially no effect on KCAT. We had no effect on the Km of ATP, but we had a very dramatic effect on the Km of microtubules. So we go under low ionic strength conditions, we have a relatively tight binding of the microtubules to KSP. As we increase sodium chloride, we diminish the affinity of the microtubules for KSP. And you may know that microtubules are highly charged, and the interactions with proteins like KSP are largely driven by electrostatic interactions. So this makes good sense, and it also points us in a direction. Perhaps monastrol and these other KSP inhibitors are acting in a way that is competitive with microtubule binding. And indeed, that's the case. This is a double reciprocal or Lineweaver-Burke plot. Enzymologists love these things. The only point here is that the convergence of these lines on the y-axis is diagnostic of competitive inhibition. And the Ki for monastrol inhibition of KSP is about 6 micromolar, and it goes up quite a bit, about 30-fold, in the presence of microtubules. So indeed, monastrol is acting as a microtubule competitive inhibitor, as are the other KSP inhibitors. Now, a simple solution to our conundrum then is that microtubule binding to KSP is influenced by ionic strength, and therefore the ability of a microtubule competitive inhibitor to compete with microtubules and therefore bind to the enzyme is influenced by ionic strength in an indirect way. It's not that the ionic strength itself is affecting the binding of the inhibitor, but rather it's setting up a situation where it's more effective in competing against the substrate. If that were true, then there should be a clear relationship between the effect of ionic strength on the Km of the substrate and the effect of ionic strength on the IC50. So this plot shows you the IC50 for monastrol inhibition in the open circles, and the Km for microtubules in the solid line. And you can see that there's quite a good relationship here, that there's a commonality. And if we use what, again, should be familiar to most of you pharmacology students, the Ching-Prusoff relationship, we can calculate what the IC50 should be, knowing the Km for microtubules at these different ionic strengths, the substrate concentration, and the fact that Ki and alpha Ki are not affected by ionic strength. And when we do that, what we would expect is a straight line relationship for a competitive inhibitor, and indeed that's what we get. So this is the IC50 versus the microtubule concentration over Km at different ionic strengths. Here are the data points, and the straight line is the theoretical prediction. So you can see that the experimental data fit very well to this. So we have a very simple explanation for what's going on here. At physiological ionic strength, microtubule interactions with KSP are diminished to a certain extent, and because of that, monasterol and these other KSP inhibitors are able to more effectively compete for the enzyme and therefore show the decreased IC50, increased affinity. So that also explains why our high throughput screening colleagues chose to run the assays at low ionic strength, because it optimized microtubule interactions and therefore increased the effective kcat over Km. So they got a bigger signal window. And there's a message there, okay? It's not only about signal. We have to think about physiology. Now, going further and looking at the effects of monasterol on its interaction with ATP, what we found was that monasterol was actually uncompetitive with ATP, and that is also true for the compounds that we have in the clinic. And we also found that monasterol acts to stabilize the ADP KSP complex, either in the presence or absence of microtubules. I mentioned to you that ADP release is rate limiting, and in the presence of monasterol, it is slowed down significantly, several orders of magnitude, both in the presence and absence of microtubules. So, to summarize the data that I've shown you so far, monasterol and the other KSP inhibitors are ATP uncompetitive, microtubule competitive. They stabilize the ADP bound state of the enzyme. And what I didn't show you in the interest of time, but we have the data for, is that these compounds have no effect on inorganic phosphate release. So they are not affecting the hydrolysis of ATP per se. What they are doing is affecting the subsequent release of ADP from the complex. And that allows us to localize the effects of monasterol and the other KSP inhibitors to this portion of the reaction cycle. They bind in an allosteric fashion, most preferentially to the enzyme ADP complex, most preferentially actually to the microtubule enzyme ADP complex. This is where the highest affinity for these compounds occur. I showed you the data for monasterol, which is a micromolar inhibitor. The same is true for our subnanomolar KSP inhibitors that are in the clinic now. So we have very effectively blocked the enzyme in this region. But there are many other ways to block an enzyme like KSP, a very dynamic enzyme. And the question comes up, what about other inhibitor modalities? And particularly when we are targeting an enzyme or any target for that matter, for infectious disease applications or for cancer, one of the greatest clinical challenges for us is the development of resistance due to mutations within the target. And so because all of the KSP inhibitors known to date share a common binding site, they would also potentially share resistance if there were mutations in that binding site. So preclinically, we have been able to use saturation mutagenesis to develop resistant mutants of KSP to all of those KSP inhibitors. And our concern was that some of these might translate into clinical isolates, and so we might want to invest in having other modalities of inhibition of this target that we could use in resistant tumors or combine with the allosteric KSP inhibitors to preemptively avoid inhibition, resistance rather. Now you might say, well, why would you want to do that? Why would you combine two drugs that hit the same target? And the answer is that has been a very effective way of preempting resistance in things like HIV. So I'm sure many of you are aware that the standard of care for HIV treatment now consists of a cocktail that includes, among other things, a nucleoside and a non-nucleoside reverse transcriptase inhibitor. So it's the same philosophy there, to hit the same target but at different binding sites to mitigate the potential for mutation-based resistance. All right, so if we're going to look for other inhibition modalities, we better screen the enzyme in a proper way. And so we went back to screen our library, and the GSK library now is in excess of 1 million compounds. So if you're going to invest in doing that type of screening, you better do it right. So we chose to look under what we call balanced assay conditions to provide the best means of identifying the broadest diversity of modalities. So we adjusted the ionic strength to near physiological conditions. The other thing we did was we adjusted the substrate over Km ratio to around 1 for both microtubules and ATP. Why do we do this? Because if one looks at the effect of substrate over Km concentration on the IC50 for different inhibition modalities, one finds that there are very dramatic differences depending on whether you're dealing with a competitive or an uncompetitive or a non-competitive inhibitor, and the best compromise between these occurs at a substrate over Km concentration of 1. So I have two motivations for showing this slide. One is to preach this to you and to make the case that this is a very important aspect of thinking about how you set up enzyme assays for screening. The other is a shameless self-promotion of my book, because that's where this comes from. Okay, as a result of changing these screening conditions, and these were not dramatic differences in screening. We had to change substrate concentrations a little bit. We increased the ionic strength. But the result of that was an enormous diversity of a variety of inhibitors of distinct modalities that we had not seen in the previous screening. I don't have time to tell you about all of them, but I am going to tell you a little bit about nucleotide competitive inhibitors that we identified through this screening. And I'm also want to make the point that whenever you're dealing with a ligand binding pocket in an enzyme or a G protein coupled receptor, you're really dealing with a conformationally dynamic system. So you don't have a target for drug discovery, you have a multiplicity of targets for drug discovery, because as the enzyme goes through its catalytic cycle, or as the G protein goes through its changes in conformation upon ligand interaction, you go through a variety of quantum mechanical substates that represent unique conformational states of that ligand binding pocket. And the point I just want to make is that each of these states represents a unique target for drug interaction. And indeed, that's the case with KSP, because the nucleotide binding pocket of KSP is conformationally dynamic, and it is quite different whether the product ADP is bound or a mimic of the substrate AMP, PMP, mimicking ATP is bound. So you see that this switch region moves significantly relative to the bound nucleotide. So we don't have one nucleotide binding pocket. We, in fact, have two distinct conformations of that nucleotide binding pocket, at least two. And these correspond to the state of microtubule binding, so that in free KSP, relative to microtubule-bound KSP, you see that the microtubule-bound state favors the binding of substrate, ATP or AMP-PMP. There's about a tenfold reduction in Ki for this inhibitor, hence a stabilization of the ATP binding form. And the opposite is true for the product-bound form. So here, there's about a tenfold increase in affinity in the free enzyme relative to microtubules. This makes good physiological sense, right? Because you want to stabilize the ADP bound form when you're off of the microtubules. When you're bound to the microtubules, you want to favor ATP binding and hydrolysis to move the protein along the microtubules. And not unexpectedly, these different inhibitors respond to ionic strength in opposite ways, again influenced by the Km of the microtubules. One question that came up in these studies is, could we combine a nucleotide competitive inhibitor and an allosteric inhibitor like monasterol and get synergy? So additivity would be fine in terms of mitigating resistance, but if we got synergy by the combination, then there might be even more value in combining two drugs in a preemptive way. And this is the equation of Yonatani and Torrell, which again should be familiar to pharmacology students, and it is a mechanism by which we can graphically determine the difference between mutually exclusive, additive, synergistic, or antagonistic binders. And what we find is that monasterol and ATP or AMP-PMP bind in a mutually exclusive fashion to KSP, but monasterol and ADP show strong binding synergy. And this translates not just to these nucleotide inhibitors, but also to some of the nucleotide competitive inhibitors that we found through screening. Now, I want to tell you a little bit more about a specific class of nucleotide competitive inhibitors, the Biaryl inhibitors, exemplified by this compound here, GSK 246-053A. We call them biaryls because a common feature of the pharmacophore is this two-phenyl ring structure. This entire class of compounds are ATP competitive. They're microtubule uncompetitive, and here are the Lineweaver-Burke plots to convince you of that. But the bottom line from this steady-state kinetic data is that the microtubule-free KSP binds these compounds with 10,000-fold less potency than the microtubule-bound form. Now, that was a major problem for us because these compounds had pharmacological properties that made them very attractive to us. We wanted to go forward with this class and look at them in cell culture and in vivo, both by themselves and in combination with the allosteric inhibitors. And to go from initial screening hits like this with Ki values in the single to double-digit nanomolar to truly potent inhibitors meant we needed to mount the medicinal chemistry effort and really optimize these compounds. Now, the best way to do that is through a combination of these biochemical studies and structural biology studies. So looking at the crystal structure of the protein bound with the inhibitors so that we know the binding site, and we can, from that, come up with ways to optimize interactions. Now, I told you this compound is ATP competitive, so what does that mean? From a structural perspective, what does it mean to be ATP competitive? Some of you say, well, that tells me where it's binding. It's binding in the ATP binding site. And try as we may, we could not rationalize the SAR for this compound series with the crystal structure by docking it to the ATP binding site. So you say, well, then do the experiment. Crystallize the protein with the compound bound. There's a problem with that. The compound is microtubule uncompetitive. What does that mean? It means that it only binds in the presence of microtubules. Microtubules are notoriously heterogeneous. Try as we might, we could not crystallize the KSP microtubule binary complex or the ternary complex of KSP microtubules in the inhibitor. So we had a conundrum here. Most of my colleagues were convinced, especially the medicinal chemists, were convinced that I was wasting my time here because it's ATP competitive. We know where it's binding. It's binding in the ATP binding site. But what we did was we decided that we would use a different biophysical method. Whenever you have a phenyl ring on an inhibitor, it offers you opportunities, if the SAR will tolerate it, to put cross-linkable groups on that molecule. And what we chose to do was to incorporate this diaza compound here, which is photoexcitable. And so the idea is that in the dark, this is a simple reversible inhibitor, and we shut all the lights off in the laboratory, and we showed that this molecule was still an ATP competitive, microtubule uncompetitive inhibitor. We took about a 10-fold hit in affinity, but still a very reasonable Ki for this molecule. When we shine light on this molecule, we go through a radical mechanism that can then cross-link to protein methylene groups, hydroxyl groups, or backbone amines. And so what we did was very simple. We ran the photolabeling study in the presence of microtubules. So we had microtubules and KSP present in solution, added the compound in the dark, photolysed. Then we ran the photolysis product out on SDS page to separate the microtubules from the KSP. We then subjected the KSP to enzymatic digestion in situ in the gel and did differential mapping with LCMS and MALDI-TOF mass spectroscopy. And by doing that, we got this mass spectrum. I won't bore you with a detailed discussion of the interpretation, except to say that it allowed us to unambiguously identify a specific site of attachment, which was this leucine residue. And knowing where it attached, we were able, through molecular dynamics, to define a binding pocket for the biaryls, which is distinct from the nucleotide binding pocket. They look proximal in this representation, but I assure you they are not. They're at about 15 or 20 angstroms away from one another, and also distinct from the common binding pocket of monastrol and all of the other KSP inhibitors. Now, there are a number of residues in this putative pocket, and one of the things that we did along with the photolysis and mass spectral data was we used saturation mutagenesis again to see if we could identify mutants that were resistant to the biaryls. And we came up with 3 resistant mutants that were distinct from the mutants that were resistant to monastrol. There was no cross resistance between those. And when we looked at the enzymatic properties of those mutants, I'm just showing 2 of them, we found that the compound, the biaryls, remained competitive against ATP, but the Ki was significantly increased. So the affinity was decreased by these mutations. When we mapped those mutations onto the crystal structure of KSP, they occur right in this binding region. So the combination of mutagenesis studies and the photo cross-linking converge to give us a common answer that this is the binding pocket for the Biaryl. So these are ATP competitive inhibitors that compete by binding to a distant site. And although there are other examples of that in biology, I think this is a novel example of, in terms of a pharmaceutical target. So we now have several locations within the catalytic cycle of KSB that we can intervene at with small molecule drugs to block the action of this enzyme. And to summarize this, we have, as I just said, shown that we can inhibit KSB ATPase activity by several distinct biochemical mechanisms. Allosteric inhibitors such as monasterol target free KSB and display higher potency under high ionic strength conditions. Monasterol and the other KSB allosteric inhibitors show synergy with ATP competitive inhibitors like ADP, but not with inhibitors of the class similar to AMP, PMP. The Biaryl compounds are ATP competitive. They bind in a unique region that is remote from the nucleotide binding pocket and also remote from the monasterol binding pocket. And the combination of these inhibition modalities may have great potential for treating human tumors individually, but more importantly, in combination, if indeed resistance becomes a major issue. So with that, I will close and thank you for your attention and take any questions.
Interpretation of the monasterol is that also operates in an allosteric fashion, presumably stabilizing the binding of ADP.
ADP, that's right.
Okay. And secondly, the monasterol has an ester group which looks like it ought to hydrolyze fairly rapidly, and yet it's the ester form that is binding.
So monasterol is not a pharmaceutical agent. It has good activity against the enzyme. It has good activity in cell culture. It does not have activity in vivo. So I used monasterol as an example because at the time that I put these slides together, I was not able to talk about the structure of the other compounds. But it really doesn't matter because the biochemical data is identical.
Yeah, the discrepancy between the in vitro and in cell data suggests another explanation, which might be an endogenous ligand that's binding to that binding pocket. So there might be some interfering substance that's produced by the cell itself.
Absolutely. So we come across this not infrequently in our research, and that's always a hypothesis. Another hypothesis is that there is an auxiliary protein that interacts with the target in a way that modulates its interactions with inhibitor. Another very common situation is that the inhibitors are slow binding. They form an initial encounter complex with the target, but then lead to isomerization of the target, which increases their affinity significantly. And if one isn't careful about how one does your enzymology, you can miss that completely. And we've seen a number of examples where we could explain the in vivo pharmacology by invoking that type of model. Basically, what we do is we look at all of those possibilities, and at some point we invoke Occam's razor and say that this is a simple solution. It's the data are all internally consistent. Does that mean it's right? Of course not, but it's a reasonable model.
So I'm very interested in your mutagenesis work to develop resistance, to then develop drugs that will combat that resistance. So first question is, do the mutants that develop resistance, are they kinetically identical? Do they work identically in the absence of the inhibitor? have you tested?
Right. We have tested it, and I'm at a loss for the details of the kinetic parameters. My recollection is that those mutants had minor defects in KCAT, but they didn't impact the KM of either microtubules or ATP. Take that with a grain of salt, though, because my memory isn't what it used to be.
Because I'm just wondering, because you have an allosteric site, and do you know what that site actually does in vivo? Is it just a hinge region, or does it actually find another molecule in nature to actually give a physiological?
Right, so that's a great question that comes up often is, are allosteric sites designed by nature for gainful employment of pharmaceutical scientists, or are they there for some other reason? And I don't know the answer to that. It is in what you would define as a hinge region. This is a very dynamic protein, and there's a lot of large-scale motions during catalysis. So I don't have a decent answer for you, Tony. We don't know. If there is an endogenous ligand, we don't know what it is.
Last question.
Well, this is a really very exciting science to explore this molecule in 3 different places, basically. Did you look on the combination of all of the 3, the ATP binding site and those 2 allosteric sites? So this is one question. And second question, did you observe any, in mice, for example, study effects on dark adaptation? Because we know that the movement of proteins, particularly in photoreceptor cells, is driven by these proteins. So do you affect that process, postponing or slowing down the dark adaptation?
So we have definitely looked at the combinations both in cell culture and in xenograft models. And indeed, we see synergy when we make the right combinations of allosteric and Biaryl inhibitors, for example. We have not progressed combinations to the clinic because so far we haven't had a need to. These things are sort of on the shelf in case we need them. And that was a business decision, and I won't go into the details of that, but it made good sense. In terms of photoperiod, we have not looked at that. It's a very interesting question, and I'm sure we'd be happy to provide a compound if someone were interested in doing those studies, but we haven't looked at that directly.